I'm Bill Andrews. I've been studying the mechanisms and consequences of the shortening and lengthening of telomeres for 30 years. I've also been studying the regulation of the telomerase gene and the role that telomeres play in the epigenetics of human cells. I've been in the business of telomere biology for a long time. I led the research that discovered human telomerase at Geron Corporation. I'm one of the inventors, co-inventors of all of the methods for measuring telomere lengths and measuring telomerase activity. And I'm one of the inventors of TA65, who I think is speaking right next door, along with a whole bunch of other nutraceuticals that are on the market for regulating telomere length. I guess I want to say that a lot of the techniques for really understanding the business wasn't really developed until years later. And I kind of feel like a lot of the companies are actually misrepresenting themselves when they talk about their products. And I don't usually market, but I'm here because I'm representing a company that I think is a very legitimate company. It's actually providing the very first telomerase nutraceutical, telomerase-inducing nutraceutical that actually works. And so I'm here to discuss the biology of telomeres and stuff like that. I'm besides working on trying to find ways of inducing telomerase, I'm also the inventor of 3 telomerase-based cancer treatments. Rytelo, for example, also called imeltistat. Telomere, which is another cancer drug for based on telomeres and telomerase, and also UV1. The bottom line is I know a lot about telomeres. I don't think you're ever going to find anybody who knows more about telomeres than I do. I've been pretty much involved in everything. But today what I want to talk about is managing your telomere lengths. Because, again, there is so much misinformation out there. So many people are being told that their telomeres are longer when they're not, okay? And I'm going to explain why that is and stuff like that. But I want to get, I believe that telomeres, controlling telomeres or managing your telomeres is the most important thing that anybody could be doing to manage their health in all kinds of ways: cancer, heart disease, Alzheimer's, and aging. So aging is just one of the things, though they all might be related to aging. And I think it's important to clear up these misunderstandings if people really want to manage their telomeres well. And that's what I'm trying to talk about today. Is I want to teach people how to really know what's going on and not listen to some people talk about things they don't really talk about. So I want to begin with talking about telomere basics, okay? And just in case there's somebody here who doesn't know what a telomere is. So what is a telomere? And what I want to do is we got to zoom in on a human being, and we keep zooming in until we get to the chromosomes. And a chromosome is made up of 2 arms, a top arm and a bottom arm. Inside of each of these arms is a long DNA molecule. DNA is like a string of beads. The beads are called bases. This is where all your genes are that give your hair color, your eye color, etc. And it's the sequence of these beads that tell you if you've got blue eyes or blonde hair or whatever. But I want you to think of this long string of beads, which, by the way, is so long it's coiled up like a slinky inside these arms. I want you to think of this long DNA molecule as a shoelace. And you know at the ends of your shoelaces you have these caps called aglets that protect your shoelaces. Well, our chromosomes, the DNA, that long string of beads that I just told you about, have the same thing shown here in yellow, and that's what a telomere is. Telomeres are the aglets of our chromosomes, just like the aglets on our shoelaces, and they protect our chromosomes. And it's wound up like a slinky, just like the rest of the DNA. Well, what I want to do is I want to unwind this slinky in the telomere part, and we find that a telomere measures about 15,000 bases in length. The average length of a whole chromosome is 100 million bases, but the telomere is about 15,000 bases, at least when we're first conceived. And this is where all the problems begin, and this is why I study telomeres, and that's because as soon as our cells start to divide, each and every time our cells divide, our telomeres get a little bit shorter. And there's so much cell division involved in going from a single cell embryo to a newborn baby that before you're even born, your telomeres have already shortened down to 10,000 bases. They were 15,000 bases when we were first conceived. Now they're 10,000 bases. This is actually hardcore science. I and others have been studying this for many, many years. The problem doesn't end here though, okay? The problem, we still have a lot of growing to do, a lot of wounds to heal, a lot of infections to fight. So there's a lot of cell division that goes, and so as you grow and get older and older, there's more and more telomere shortening. And when telomeres get down to 5,000 bases, our cells enter a phase called senescence, and we die of old age. Our cells at that point lose the ability to function, and we die of old age. Let me just go over that one more time. We're conceived at 15,000 bases, we're born at 10,000 bases, and we die of old age at 5,000 bases. This is an actual ticking clock that we all have inside of us. And it's a very, very well understood clock. We know everything from the very beginning of how it works to the very end of how it works. It's the only thing in aging that is that well understood. And I want to say that no matter what else we do to try to cure aging, you can hear a lot of people talking about trying to do things that cure aging here. No matter what else we do, aging is never going to get cured unless we solve this problem too. Because when you do the math on telomere shortening, what I call the basal level, and I'll come back to that in a little while, there's a minimum, you can slow it down a little bit, but there's a rate that you can't slow it lower than. When you do the math on that, it turns out that we all have a theoretical maximum lifespan and health span of 125 years. And that's only if you have the perfect genetics and lead the perfect lifestyle, because a lot of things that you can do, including your genetics, can accelerate that shortening, limiting your lifespan even shorter. And nobody has lived to be 125, at least in recorded history. And there's people all talk about Methuselah and stuff like that, but there's a good chance that they were counting years by full moons. And it's so really this is a limit we have on our lifespan, and this is because of this, and because we have to solve this problem if we're ever going to cure aging. This is why I've been devoting my last 30 to 40 years of my life to trying to figure out how to solve this problem. Okay, so one of the misconceptions that is spread a lot, you hear all the time, is that telomeres shorten. They don't actually shorten. Okay, this is a misnomer, but it leads people to start thinking there's different things they've got to do to prevent the problems with shortening. I call it shortening, and you're going to hear me say it a lot of times, but they don't actually shorten in most cases. You can do things that cause them to shorten. They also don't erode, unravel, all these words you hear, shred, degrade, wear away, break off, get damaged, etc. Now, that said, you always hear about people saying, you can repair your telomeres. Well, they don't get repaired either because they didn't get broken to begin. You can't fix what's not broken. Okay, so it's not what's going on here. This is a totally separate issue than DNA damage and DNA repair. And the reason why I have a brick wall here is because I want to use this brick wall as an analogy to explain what's really going on. Okay, so every time a cell divides in our body, every time a cell divides and to make two daughter cells, everything inside that parent cell has to first be duplicated so that when the cell divides, the two daughter cells have the same exact components that the parent cell had. So I'm going to use this brick. We're going to make a new row of bricks on a brick wall as a way of explaining DNA replication. So I'm going to get rid of the other bricks and get rid of that cat. And now the cell's about to divide, the DNA gets replicated, and we have a brick layer laying a new row of bricks. This brick layer is called DNA polymerase inside of our cells. And this analogy is so accurate, it's amazing that I can use this. Okay, so what's happening now is we got to look at what's happening at the very end of the chromosome. And as you see, because the brick layer is on top of the wall, there's going to be a problem. And that's he can't lay a brick in the last place that he was standing. And this is called the end replication problem. This is what we've known about in DNA forever. When DNA replicates, the cell doesn't have the ability to replicate all the way to the very end. It can't replicate the very end. It's referred to in the scientific literature as the end replication problem. So it's shorter. The original DNA is still there, still long, but the new DNA is shorter. So the cell is going to divide again. Again, that brick layer is going to fall off the wall, and the DNA is going to get shorter and shorter and shorter over and over again. I call this, as I mentioned before, basal level telomere shortening. And there's very little that we can do about this. Now, if you are really impatient and you want to accelerate your aging, there are things you can do to do that. And so I said telomeres don't break or degrade, but you can if you really try hard. And that's anything related to an unhealthy lifestyle, like obesity, psychological stress, smoking. All these things will generate things like free radicals, inflammation, and stuff like that, which will very much accelerate. I call this accelerated telomere shortening. And these are things that we all do. That's why none of us live to be 125, because we all do something that's causing accelerated telomere shortening. But there are other things we can do. So accelerated telomere shortening, we can reduce that to the maximum by reducing inflammation, oxidative stress, UV exposure, etc. Let me just give you some examples. Antioxidants. So this is why you want to be taking antioxidants, anti-inflammatories. Don't smoke, don't be obese. Avoid toxins, proper exercise. Bottom line, exercise as much as you can, but keep it fun. As soon as it quits being fun, quit. Wait until you feel you can exercise again. If you take that kind of philosophy, you'll find out you can start exercising all the time and never get tired, and it's not bad for you. The people that exercise overdoing it can accelerate your aging if you really push it and you're an occasional exerciser. And so proper exercise. Manage sugar levels. Sugar levels are very inflammatory, and they cause a lot of oxidative stress, which will accelerate telomere shortening. So find a way to really manage your sugar levels. Avoid inflammatory foods. Reduce psychological stress, the kind your boss gives you, that can accelerate your aging, etc. This is where I want to introduce an analogy that I think is very appropriate for talking about telomere shortening and aging and practically a lot of diseases. And that's that normal aging and declining health is a one-sided tug-of-war. Okay, so we only have people pulling to shorten your telomeres, nothing to offset that in our normal lives. So every time our cells divide, our telomeres get shorter and shorter and shorter, with nothing stopping that. Shortening that I just mentioned is a combination of both the accelerated and the basal level telomere shortening. But we can, as I just mentioned, get rid of the accelerated telomere shorteners and get down to basal level telomere shortening, and that is going to slow down the rate of telomere shortening. And that's really important. So the best you can do to get rid of the accelerated telomere shorteners, the better. Question is, how do we slow it down even further? And this is where I got really involved in the business 40 years ago. It turns out telomere shortening doesn't occur in our reproductive cells. And nobody knew why, but it was obvious that they couldn't be shortening, because if they were shortening, our children would be born with shorter telomeres than we have. Their children would be born with shorter telomeres than they have. And we would have been extinct as a species a million years ago because we wouldn't have any DNA left. So when I first got into this business, I was trying to figure out, why is it that our reproductive cells don't have telomere shortening? And so in our reproductive cells, we do have people on the other side of the tug-of-war. It is a two-sided tug-of-war. So we have shorteners and lengtheners, and somehow it's a tie. The tug-of-war is a tie in our reproductive cells. There is no telomere shortening. Well, in looking at this and studying this, I ended up leading the team that discovered human telomerase back in the early to mid 1990s. This is the DNA molecule shown here as a double helix, and telomerase is shown as a factory here. It binds to the tip of the chromosome and it lengthens the telomeres. So the telomeres still shorten, but telomerase then re-lengthens them. Okay, so we have, again, going on here, the lengtheners are telomerase. That's the only thing that's ever been discovered in human cells that can lengthen telomeres, and that's this enzyme called telomerase. So if we go back to the brick lane analogy, the bricklayer is still going to fall off the wall because I said it's two different processes. There's shortening, and like an angel, telomerase comes in and replaces that lost brick. And we have found from experimental work in a petri dish that if we add more angels, the angels start adding bricks and making them even longer, which opens the door for eventually reversing aging through telomere biology, which is my goal in life, to figure out how to do that. Okay, so how am I doing this? So at my company, which is Sierra Sciences in Reno, Nevada, we are looking at the telomerase gene. This is the blue thing here is the gene for telomerase. The gray bar is the chromosome, this long string of beads. It extends a mile in each direction, but I'm not showing all of it. Next to every gene in our body, there's a regulatory element, which is like a dimmer switch, which turns genes on and off. In our reproductive cells, that regulatory switch is on. As a result, in our reproductive cells, the gene produces telomerase. Now, for weird reasons, in every other cell of our body, there's a protein that binds to that regulatory element and shuts the gene off. Now, my Ph.D. is in molecular and population genetics, which means I study the molecular biology of DNA and gene expression and things like that, but I also study the evolution, which is what population genetics is. So if anybody wants to know why we evolved a way to shut this gene off when it sounds like it's a terrible thing for us to do because it causes us to age, go watch my YouTube videos because it takes me an hour to explain this. But it essentially comes down to the fact that eliminating us after we've raised our young is actually beneficial for the survival of our species because it increases diversity. If you understood that, that's really good. But you can go listen to videos where I explain that in a lot more detail. But we're stuck with it, okay? We have this telomerase gene turned off in every one of our cells, and that's why we are aging. Okay, so what I'm doing at Sierra Sciences is, me and my team, we're looking, oh, by the way, that's called a repressor. We're looking for anything that will bind to that repressor, dislodge it, and turn the gene back on again. So we've been doing that for 30 years, and a lot of things that you've heard about that induce telomerase were discovered by doing this particular assay. Now, so I want to key in on this word expression. I use it a lot, but this is what expression is. Expression is turning on a gene. So in the field of molecular biology, we talk about gene expression. Gene expression is turning the gene on, and that's what we want to do in order to get anything to actually work with the case of telomeres and telomerase. This is a picture of our robotics lab. There's 2 robots in this picture. There's also 2 humans. And these robots would test everything for us, mostly plant extracts nowadays, looking for any kind of plant extract that will, when combined with the cells, will induce that cell to start to cause that repressor to come dislodge from the regulatory element, turn the gene on, and lengthen telomeres. So that's what we do. We can test up to 4,000 different plant extracts a day, and we've done a lot. Something, again, we're focusing on the mRNA. For those of you that know about gene regulation, the very first thing that happens when you turn a gene on is an mRNA gets created. And it turns out with the telomerase gene, the mRNA is the only rate limiting step to actually getting telomeres lengthened. So we measured that, and it turns out there's a really strong, we've discovered over and over again for 30 years, there's a 100% correlation between the amount of mRNA for telomerase and the length of your telomeres. So this is the best and by far the most sensitive assay that exists for looking at the production of telomerase. And this is why we use this assay. The success that we've had has come from actually finding people to put on the other side of this tug of war. Okay, so we have tested a bunch of things. We have found people, and now it is, again, telomerase, but notice that we haven't won the tug of war in this picture. We still have that one person pulling against 4 people pulling to shorten, but at least we are slowing down the rate of telomere shortening and decreasing the rate of aging. And so it's still, there's a back and forth because of the fact that there is still lengthening and shortening as two separate processes in the cells. Now, even though it hasn't reversed aging, this is a good thing, okay? And believe me, everybody who claims that they have lengthened telomeres, they have not. But nothing has slowed it further down than the things that we have discovered that I'll be talking about in a few minutes. So we have tested close to 20,000 different plant extracts right now. We're calling them nutraceuticals here. Sometimes they're complete extracts, sometimes they're purified things from extracts. And we have found over 40 different plant extracts that will induce the production of, or expression of telomerase. And this is, so we have this, but what we can do even better is by reducing the accelerated telomere shortening, by eliminating some of the people, we can even make things better. Now, the question comes up, is there a nutraceutical where it actually produces more people on the right side of the tug of war, on the left side of the tug of war? And even though there's a lot of people, including I think the ones next door, that are saying that they can do this, nobody can do this yet. But that is my goal in life. That's what I'm reaching for. And I'm going to continue looking, doing the research until I find something that does it. But it does not exist yet. Okay, so that brings up to the next misunderstanding that's going on in the whole field that I think is the most critical one and that I want to spend the rest of my time talking about. And that is, why are many scientists and companies and organizations claiming to have products that lengthen telomeres? What's going on? I'm not going to say they're lying to you, but it might be that they are totally, so the question is, do they really? The question is, they might just not understand the telomere biology as well as they should. The problem is, the average telomere, this is a really key point of the whole talk, the average telomere length in blood or saliva, because you can do telomere length testing in blood or saliva, can become longer without telomeres ever being lengthened. And this is what a lot of the scientists don't seem to understand, okay? But they haven't been working on telomere biology as long as I have. In order to really demonstrate that you are lengthening telomeres, you have to also demonstrate that you are demonstrating telomerase expression. You have to show that because, as I said, the only way to lengthen telomeres is to have telomerase expression. And if you don't have telomerase expression, you do not have telomere lengthening. The telomeres are appearing longer for some other reason, which I'll come back to shortly. Now, to demonstrate that you were actually winning the tug of war, you are producing so much telomerase that you are winning that tug of war, you have to do some really great tests to show that you have very high levels of telomerase expression. And that's something else that a lot of people don't seem to understand. They think you produce any bit of telomerase, your telomeres are getting longer. That's not true. You're just slowing down the tug of war. So let's come back to this first point, because this is where I want to get into some detail here. Blood is a very mixed population of cells, all kinds of cells, but what's more important, it's a very mixed population of different telomere lengths in the cells of the blood. So this is showing that if you take a blood sample, you have blood cells that have short telomeres, some that have long telomeres, some that have average telomeres. The reason why there's such a variety of telomere lengths in the blood is because different cells in the blood have had a different history of the number of times that they have divided to be in the blood where they are. So you have a whole distribution of these. Okay, so imagine you could reach in and remove the cells that have short telomeres. What's going to happen to the blood? You're now going to remeasure the average length of the telomeres, and you're going to find that the average length is longer. Not because they got lengthened, only because you removed the cells with the short telomeres. The analogy that I like that you often hear about is if you have a company and you fire all the dumbest people in your company, the average IQ of that company increases without anybody getting any smarter. Okay, the same thing is happening here. You're removing cells and the average telomere length gets increased but no telomeres got longer. And that's something that a lot of these other companies don't understand. Okay. Likewise, if you add new cells to the blood that have long telomeres, again, the average length of the telomeres increase, just as if you had hired a bunch of geniuses in your company. The average IQ of that company increases without anybody getting any smarter. Okay, getting that concept out of the way, let's dig into this a little bit deeper. This is a publication here where you can actually go, and if anybody wants to learn more about this, it turns out that cells with short telomeres are more sensitive to toxins than other cells are with longer telomeres. So when there's a toxin administered to the blood, the cells with the short telomeres are going to be preferentially eliminated. Okay, so if you hear about a product and they say that their average telomere is being lengthened, you got to question, maybe their product's a toxin. Okay, maybe it's actually getting rid of the cells with short telomeres because it's killing them off. Okay, so that's one thing. The average telomere length is again going to get longer just by killing off the cells with the shorter telomeres from the toxins. Likewise, and this is another publication to show what I'm about to say now. If your treatment that you're providing the patients or yourselves, is it all immunogenic? You are going to induce dormant immune cells in the bone marrow or elsewhere. They're going to be like memory cells or naive immune cells that are going to divide and migrate into the blood. And because these are dormant and they're essentially young, even though you might be an old person, their telomeres are long, and so they are, when they enter the blood, they're providing new cells to the blood with longer telomeres. And again, this is going to cause the average length of the telomeres to be longer without increasing, without being lengthened. And this is a real problem. So you're going to see a lot of people showing before and the after is showing longer telomeres. And the key that I seem to be sometimes the only person that seems to understand outside this room is that a treatment that is toxic or immunogenic will do the same thing as this without any lengthening. And that's what you got to be really careful. Okay, so the solution, what they have to do to convince you that they have something that really lengthens your telomeres, is they have to show the way that they measure telomerase expression and show that they're actually getting telomerase expression to occur. Okay, someone tells you that their treatment lengthens telomeres. Before you buy their product, I want you to ask if their treatment induces telomerase expression. If not, it does not lengthen telomeres, period. If they say, well, they didn't do the assay or didn't do the test, then ask, well, why not? It's not that hard. If they're claiming that their product lengthens telomeres, it must be expressing telomerase. Why aren't they measuring telomerase? And what you're going to find is that, well, we did try it and we couldn't get it to work. And being one of the inventors of all these procedures that measured telomeres and telomerase, I know that a lot of people like to take shortcuts and stuff like that and get wrong answers, but this is the problem. If they didn't measure it, walk away, okay? Now, HeLa cells. HeLa cells are a cell, when I was still working at Geron, where we discovered human telomerase, we discovered that HeLa cells express just enough telomerase to tie the tug of war. This is a very useful cell line because it gives us a standard to measure against. So it doesn't produce so much that it lengthens the telomeres, it produces just enough to keep them from shortening. So we know that if we could find a telomerase inducer that produces just as much telomerase as is found in HeLa cells, we know that we would have something that stops telomere shortening and stops aging. If we could find something even higher, then we know that we are now lengthening telomeres and possibly reversing aging, which we won't know until we actually achieve doing that. Okay, so now, if someone tells you that their treatment lengthens telomeres, ask if their treatment induces more telomerase than HeLa does. This is not something that everybody doesn't know. Everybody knows this. In fact, these companies have people that used to work with me that were part of doing these studies where we learned all this stuff. If they If their product doesn't produce enough, then it doesn't lengthen telomeres. But if it does, if they can show that it does produce more telomerase expression than is found in HeLa, then they do lengthen telomeres. If they say they didn't do the test, again, say, why not? I mean, it's not hard to do. These are things that these companies should be doing if they are claiming that they are lengthening telomeres by inducing telomerase expression. And this is a real problem. Okay, so like it or not, we test everything in our assay. And because our assay is actually measuring telomerase expression. We have the best assay in the world, though everybody has access to it, we just invented it. If we tell them our results, so when they tell you that they have a product that lengthens telomeres, ask them if they have the Sierra Sciences or Bill Andrews seal of approval. Because if they don't believe them, okay? And we have tested it and we have told them yes or no. Okay, so this is just some data now. We have tested every single nutraceutical, even the ones that I invented, before we had the great assays that we have now. We have tested every one of them, and none of them produce telomerase, express telomerase. I'm not going to mention who they are. I don't want lawsuits or cease and desist letters. But we've also we tested diets. There's a lot of diets that claim to lengthen telomeres and induce telomerase. None of them do, not a single one. We've tested lifestyles, including psychological things like meditation and other things that people are claiming will lengthen telomeres. None of them cause telomeres to be expressed. We've also looked at therapies. You've been hearing a lot of therapies. People will get into this chamber or do this treatment or hook up to these electrodes and your telomeres will get longer. None of them produce telomerase expression. Okay. Through our assays, I've already alluded to this a little bit, we have absolutely found a few that do into plant extracts that do induce telomerase. That's shown here. Turmeric without curcumin. Curcumin actually inhibits telomerase. So don't take curcumin at the same time you're taking turmeric if you want to lengthen your telomeres. Pomegranate, some other things, hazy, galnut, I can't read from the angle here, but all of these things we have seen induce telomerase expression, with turmeric being the strongest. All of these things, and this is, I mentioned before, I'm pissed off at a lot of the claims that all the companies are making. I don't usually market stuff like that, but I'm supporting this company that has now combined all these things into a product called Telo-Vital. The company's name is Touchstone Essentials. This is the first product that I feel really proud of, that I played a role in developing, because it really does induce telomerase expression. Nobody's making claims that it lengthens the telomeres. It doesn't win the tug of war. It does, though, decrease the rate of telomere shortening in this tug of war better than anything else that exists in the nutraceutical world. Okay, so it's not as much as HeLa, but nobody is, and this is still a really good thing that it actually will slow down the rate of telomere shortening. It also has things in it that will prevent what I was talking about before, accelerated telomere shortening. So it does remove some of these people. So you got the best of both worlds. That's even better, having a reduction of the accelerated telomere shortening, the maximum of anything you'll ever find anywhere for actually lengthening, putting people on the other side of the tug of war. And so this is great. Now, I'm available for questions. I'm finishing talk now, the Q&A, if anybody has any questions. Q&A is always my favorite part because I know all the answers. And so I will be available until everybody else is done. So I want to say thank you very much. My motto is cure aging or die trying. I'm not stopping now. All right, thank you. Any questions, or did I already answer them all? Okay.
Bill, if you go back to the slide where you've got the 2 against the 1, when that shortening is happening and you're kind of doing this tug of war, each of those losses goes through some stage of the body, right? Do you get a little bit of that back, or how does it affect the body when that tug of war is happening?
Well, it's really just slowing down the rate of telomere shortening. You're not feeling any tugging inside your cells. But so without this person here, the shortening would be at one rate. By having this person here, it's just slower. So you are slowing down your aging. Okay. Now, there's no way to measure the slowing of aging. That's impossible. You can measure it if you have 1,000 mice. You can measure aging in mice. But I'm going to tell you, mice do not age by telomere shortening, contrary to popular belief. Okay, it's a very, telomere biology in mice is extremely different from humans. The only animals actually, so there's a lot of stuff that's going to happen with the pet world too, because it turns out there's very few animals in the world that actually age by telomere shortening. But fortunately, dogs, cats, and horses are 3 of them. And so we're planning on looking for things where you can treat your pets too, but it's not going to work on your pet mice or your pet rabbits. Rodents do not age by telomere shortening. All right, any other questions? Yes?
Are you studying the HeLa cells?
Is there anything left to study in HeLa cells? We know so much about it. The entire chromosome is sequenced. We know that it has the telomerase gene in it 7 times. So when telomeres, this is a really important thing, because HeLa is a cancer cell. And I believe that, and I actually was National Inventor of the Year for my cancer research. So I'm not just aging. I study all aspects of health, and a lot of the products that I've discovered, a lot of big biotech breakthroughs had nothing to do with aging. But cancer is one of the things I've done a lot of work on. And it turns out one of the major causes of cancer for a healthy person is critically short telomeres. When telomeres get really short, it induces mutation rates. Mutation rates skyrocket. You can actually, from me looking at cancer cells in the microscope and looking at the chromosomes, I can see the mutations. They're so big. They're chromosome rearrangements and stuff like that. So we want to keep telomeres from getting short because that causes cancer. So HeLa is so rearranged, we can see the telomerase gene reproduced, doubled 7 times in the chromosomes at different places, and we still don't know exactly what is causing it to produce the telomerase, but it's like you have a gene, you have a telomerase gene that has that regulatory element shutting off the gene. Well, if all of a sudden you break in between those two and rejoin something else, you can put an on switch in front of the telomerase gene, and that's what HeLa does. But did you have any particular questions about HeLa?
No, just if you figured out why you're producing so much. Like, can you mimic?
We have really good guesses from analyzing the DNA sequence, but the simple fact that we know it's hard to tell which gene is actually the one producing the telomerase, because once the mRNA gets made, it's released from the DNA, so you don't know which gene it came from. So it's pretty hard to do. But HeLa cells, for those of you who don't know, is from a woman named Helen Lax. That's not her first name, but Henrietta Lax, sorry. They used to say her name was Helen Lane because she was keeping her identity. But then when they started realizing that these cancer cells were becoming so useful in medicine, her family came out of the woodwork and said, oh, it was our ancestor, Henrietta Lax, that had cancer, and they got the cells from her. What other questions? Anything else? Yes?
Telomere itself contain any useful information, or is it just as a protective?
Does telomerase itself contain any
The
information?
telomere.
Oh, the telomere. The telomere doesn't not code for anything. I mean, there are a lot of papers that publish. What I do is something called critical meta-analysis of peer-reviewed studies. So I read the studies, and I don't believe them all. A lot of them are really, let's say, incomplete or inaccurate. There are a lot of studies claiming that telomeres do produce an RNA called TERA, T-E-R-R-A. And the claims go beyond that to say that these TERAs then regulate genes on the chromosomes. And I have not been convinced of that yet. But right now, there is no coding sequence in telomeres that will allow a protein to be made. There are too many like stop codons and things like that in it. So telomeres, all the telomeres are like a cap on your shoelace. Now they do have sequences that are very similar to the sequences, this is going to get too scientific, sequences found in enhancer sequences. Okay, an enhancer in a chromosome is a piece of DNA that's a long ways away from the gene that will have what are called transcription factors that will bind to that enhancer. Then the DNA folds over, comes in contact with other promoters, genes, and turns genes on or off. Okay, telomeres have every hallmark of an enhancer. So, and we have data to support this, but I believe that we're going to find conclusively that telomeres are enhancer sequences. Transcription factors will bind to the sequences in the telomeres. The DNA then folds over, comes in contact with genes, turns them on and off. But the shorter the telomere gets, the less able it is to reach those genes. That's why we age. Did that answer your question? Okay, probably more than you needed. Any other questions? He knows everything. I don't know what this question's going to be.
So when telomeres get short, bad things happen.
Yes.
When we age, gray hair, wrinkles, lost memory, poor eyesight, muscle atrophy, skeletal atrophy, low energy, weight gain. You work with other companies.
You
that have-
just listed about 1% of all the problems, yes.
Yeah.
Go ahead.
So you work with other companies. You created many other products. What have you witnessed yourself on the transformation when we support the?
Okay. So here's the thing. So I say that putting one person on this side of the tug of war isn't sufficient to lengthen telomeres. We did not set this up, but this is, I think, something I'm glad he asked. It turns out that the shorter a telomere is, the easier it is to lengthen. Okay, now if you read my books or watch some of my videos, I describe this as the fact that a telomere is not a linear thing. It's rolled up like a ball of yarn. And the tip of the telomere is in the middle of that ball of yarn. So the longer the telomere, the more buried the tip is, and the harder it is for telomerase to get to that tip to lengthen it. So when the telomeres get really short, it's easy for the telomerase to get to that, and then it gets lengthened. So if you are over 50, and I can tell most of you aren't, if you are over 50, you probably have sufficient numbers of what I call critically short telomeres. And that even having one person on the other side of the tug of war will probably be enough to lengthen, physically lengthen, make them longer, the shortest telomeres. And you will possibly see age reversal. And this has happened with some other nutraceuticals that I discovered, where we did test them and we did see vision getting better, endurance getting better. My favorite subject, endurance. My wife and I are endurance runners, so we get a kick out of it, seeing anything that improves endurance. But in some cases, people's hair color came back, but not everybody. And so I always say, well, if your hair color changed because of telomeres getting shorter, then your hair color come back by lengthening the telomeres. But in a lot of cases, the hair color changes or hair loss has nothing to do with telomeres. In fact, some people, I've got a whole 5 minutes left, okay, which is way more than I thought I had. Okay, so some people lose their hair because of telomere shortening. If you lengthen your telomeres, your hair will come back. It's theoretically, at least that's how I can, the only way I can explain why I have seen unambiguously some people's hair come back when they started taking nutraceuticals that I discovered. Did that answer your question, or did you want to add to that? Because you already know all this. He's just trying to get.
I truly believe this is the holy grail of anti-aging. And there are so many, the body has an innate ability to heal itself. And I've never seen any other scientific breakthrough that has, with the research. So,
when I'm at the booth answering questions, I guarantee he'll be there too. You can ask him these kind of things. I don't get that much into anecdotal data. I try to focus on the science, but you can listen to him because he knows all the anecdotal data. Yes. That's Sunny Wynn. Thank you very much.